Okay, so first we'll talk about the spatial senses, then we'll talk about the endocrine system. So, the sensory system includes two things. Sensory system includes two things. One is called somatosensations. Okay? And another part of sensory system is called spatial senses. Okay? So those are the two parts of the sensory system in our body: somatosensations and spatial senses. Somatosensations are pain, touch, temperature. Those are the main somatosensations that you feel everywhere in the body. You feel everywhere in the body: pain, touch, temperature. Those are somatosensations. Spatial senses include vision, that means sight, audition. Audition is hearing. Gustation. Gustation is taste. And olfaction. Olfaction is smell. Okay? So these four belong to spatial senses: vision, audition, olfaction, and gustation. Now, you see the main difference between somatosensations and spatial senses is, in case of somatosensations, the somatosensory receptors are present everywhere in the body. Make sense? You have pain, touch, temperature receptors present everywhere in the body. It's not localized. But in case of your spatial senses, the receptors are highly localized. For example, your visual receptors are not present everywhere in the body, only in the retina of the eye, in a very small structure. Your hearing receptors are present inside the ear in a very small structure. Gustation, taste receptors are present in the tongue. Smell receptors, olfactory receptors are present inside the nose. So, for spatial senses, the receptors are present in localized organ areas of the body. But in case of somatosensations, everywhere in the body. Anyway, so we'll talk about some spatial senses. We'll talk about audition, olfaction, and gustation today. So, olfactory system is responsible for olfaction, smell. Gustatory system is responsible for taste, perception. And auditory system is responsible for hearing or audition. So we'll talk about those three systems. First we'll talk about the olfactory system. We'll talk about the parts of the olfactory system, how the signal transduction occurs in the olfactory system, and olfactory pathway. That means how the smell signal goes from the nose to the brain. That's the pathway, right? So we'll talk about that. Then we'll talk about the gustatory system. Same things we'll talk about in gustatory system. We'll talk about the parts of the gustatory system, then the transduction of signal and pathway of signal. How signal reaches from the tongue to the brain. And then we'll talk about the auditory system. Again, the parts of the auditory system and auditory pathway. Now, one thing before we start, you need to remember that all of those three systems have two main parts: the peripheral organ and the central part. Central part is located in the central nervous system, and the part which is called the peripheral part, that is outside of the central nervous system. So all those three systems have peripheral part and central part. Central part is located inside the central nervous system, and peripheral part is outside of the central nervous system. Number one. Number two, another thing you need to remember, that the receptors are always located in the peripheral organ. So receptors are located in the peripheral part or peripheral organ, not in the central part. Peripheral part is responsible for receiving the signals. So peripheral part, I am repeating again, peripheral part is responsible for receiving the signals, and central part is responsible for processing the signal. So that will be the simplest way to say: receiving and processing. Peripheral part will receive the signal and send it to the central nervous system, and central nervous system will do what? Will process that signal. So we'll talk about the olfactory system first. Olfaction is the smell. Smell is the olfaction, and the parts of the olfactory system, peripheral part and central part. And in case of the olfactory system, the peripheral organ is the nose. Nose is the peripheral organ. And inside the nose, you have olfactory epithelia. In the innermost layer of the nose, you have the epithelial lining inside the nose, and the receptors are located in the olfactory epithelium. So inside the nose, nose is the peripheral organ for olfaction. Inside the nose, you have an epithelial lining that is called olfactory epithelium, and the receptors are present in that epithelium. That thin layer contains the receptors. Central part is located inside the brain, inside the central nervous system. We'll see what are the parts there. So here you see inside the nose, you see one half of the nose, and this is the nasal cavity, and the epithelial lining is here, innermost lining, and where the receptors are located. Okay, so nasal cavity is a pretty large cavity. The receptors, just remember that receptors are not present everywhere inside the nasal cavity. Receptors are present in the olfactory epithelium of the roof of the nasal cavity. Roof means the upper part. Okay? In the upper part of the nasal cavity, the receptors are located in the olfactory epithelium. And from the receptors, the signal is taken to the olfactory bulb. So receptors are located here in the epithelium. From the receptors, the signal is taken to the olfactory bulb. Who takes the signal? The olfactory nerve fiber. So there are many olfactory nerve fibers. They will carry the signal from the receptors here to the olfactory bulb. So these are the olfactory nerve fibers. And then from the olfactory bulb, the signal will go to the higher brain structures. Okay, so we'll see those structures later. Just know that from the olfactory bulb, the bundle of fibers, that is called olfactory tract. Tract, you know that tract is bundle of fibers. So from the olfactory bulb, olfactory tract will carry the signal to the higher brain structures. Okay. What happens when you smell something? For example, you have a cake in front of you here. From that food, the smell molecules, they float in the air. Smell molecules must be able to fly in the air. So they fly and enters into your nose in air and with the air, and then binds with the receptors. So if this is the receptor in the olfactory epithelium, and this is the odor molecule, smell molecule that is floating in the air, will enter into your nose and will get attached to the receptor like this. Okay, and when that binding will occur, the receptors are everywhere here in the roof part. So when the odor molecules will enter into the nose, will bind with the receptors, then the signal will start to travel and will go to the olfactory bulb. So one important property of odor molecules is they must be able to float in the air, otherwise you won't be able to smell. They must be able to float in the air and enter into your nose, right? Otherwise you won't be able to smell. So that's an important property of odorants. Okay. So from the olfactory bulb, olfactory tract takes signal to higher brain structures. Which structures receive signal from the olfactory bulb? 4 different structures in the brain: olfactory cortex, hypothalamus, amygdala, and limbic system. So from the olfactory bulb, olfactory tract will take signal to those 4 structures. Okay. Remember here, you know that thalamus is called the major sensory relay station, and I told you before that almost all sensory signals must be relayed in the thalamus, but I said almost all, not all. This is one sensory signal that will bypass the thalamus. That means it will not be processed or relayed in the thalamus. It will directly go without entering into the thalamus. So if I ask you which sensory signal will not be relayed in the thalamus, that's the olfactory signal. Okay? It will bypass the thalamus. But almost all sensory signals are relayed in the thalamus. So you see here, if you magnify the olfactory epithelium, this is the innermost epithelial lining of the nasal cavity, that's the olfactory epithelium. And if you see under the microscope, 3 types of cells are present in the olfactory epithelium. How many? 3 types. What are those? Main cells, as I have mentioned, the receptors are located in the olfactory epithelium. So receptors are the main cells, receptor cells, olfactory receptor cells. there are two other types of cells are present. One type is called supporting cells, and the other type is the basal cells. So those are three types of cells present in the olfactory epithelium. I am repeating again: the receptor cells, supporting cells, and basal cells. The most important ones are receptor cells, because receptor cells will send signal to the olfactory bulb, right? And supporting cells just support the receptor cells, keep them in right locations. So they just support the receptor cells. Basal cells are also very important. Basal cells are stem cells. Okay? So basal cells are stem cells. That means what? They can produce new cells. Stem cells, always remember, stem cells in our body are the cells. They can produce new cells. So basal cells are important because you know that inside the nose, the chance of infection is very common, because we inhale the air directly from outside. We take the air inside the nose. So infection of nasal cavity is common, and microorganisms are always entering there. So they can kill the receptor cells, and they do actually. All the time we lose the receptor cells inside the nose. So therefore we need the stem cells to be there. So when the receptor cells die, stem cells will produce new receptor cells. So that's why having the stem cells is important inside the nose. There. So just remember, if I ask you what are the three types of epithelial cells, olfactory epithelial cells, those are three types, and which one is the stem cell, which one is the main cells, you need to know. Okay, now if you see a receptor cell, this is a receptor cell, the yellow one, and these are the basal cells, blue ones, and red ones are the supporting cells. Now, if you see the structure of a receptor cell, it is like a neuron. It is a neuron-like cell. That means it has a cell body, a long axon, and a dendrite. Now, the end of the dendrite, if you see here, this is the cell body, long axon going to the olfactory bulb, that's the olfactory bulb, and this is the dendrite. Okay? Now, if you see the end of the dendrite, you'll see many cilia, soft hair-like structures. So these are the cilia. And on the cilia, you have the receptors. These are the receptors. So receptors are tiny protein structures located on the cilia. Okay? So two things here, don't get confused. One is, this whole thing is the receptor cell. This is one thing. The whole thing is a cell, and what type of cell? Receptor cell. Why this is called a receptor cell? Because it has receptors. So receptors are very tiny protein structures located where? On the cilia of the receptor cells. Okay? Yeah, tiny protein structures located on the cilia of the receptor cells. Okay, So basically these tiny structures, although tiny, if you see under a very powerful microscope, these tiny structures are very nicely structured, like as I have shown you there. This is a receptor, another receptor could be like this, okay? So the odor molecules, like if the shape of the smell molecule is like this, it will fit here. If the shape is like this, it will fit here. I'm not going to talk in detail about that, but know that these receptors are protein structures and they have binding sites on them. Anyway, so now the long axons of olfactory cells are called the olfactory nerve fibers. So basically, olfactory nerve fibers are the long axons of receptor cells, and they will go to the olfactory bulb. They will take the signal to the olfactory bulb. And inside the olfactory bulb, you have the mitral cells. So these are the mitral cells located inside the olfactory bulb. I may ask you that what type of cells you will find inside the olfactory bulb. Those cells are called mitral cells. And the mitral cells will receive the signal from the olfactory nerve fibers, okay, or axons of the olfactory receptor cells. And then the axons of mitral cells will bundle together to form the olfactory tract. Okay, so olfactory tract is the bundle of mitral cells, the bundle of the axons of mitral cells. Okay. So now I am going to repeat it. You have the receptors here on the cilia, and when the odor molecules will enter into your nose, they will bind with the receptors here on the cilia, and when that binding will occur, then the signal will start to travel. This is dendrite, so it will take the signal towards the cell body, and from there the signal will get out to the axon of the olfactory receptor cell and will go to the olfactory bulb. In the olfactory bulb, you have the mitral cells, and the dendrites of mitral cells will receive that signal towards the cell body, and from there the axons will take the signal out. And the bundle of the axons of mitral cells are called bundle is called the olfactory tract. Now another thing, just remember here, from the olfactory tract, the signal goes to these four brain structures I have mentioned before. The first one, the olfactory cortex, is responsible for the perception of smell. Like what kind of smell is that? Is it the smell of a rose or a banana or a mango? Okay, so that is the perception. That's the olfactory perception. What kind of smell is that? That is given by the olfactory cortex, or processed by the olfactory cortex. Other three structures, hypothalamus, amygdala, and limbic system, those three structures are heavily involved in olfactory memory and emotion. So those three structures are related to olfactory memory and emotion. What does it mean? You know that if you have smelled something in your very small, in your childhood, only once in your lifetime, sometimes after 20, 30, 40 years, if you smell second time, you can tell that some time in my life, this smell seems familiar. to me. Sometime of my life I have smelled that. Olfactory memory is one of the most powerful memory in our brain. Make sense? And like every time I think that I will go to watch a movie, I go to movie theater, I start to smell popcorn. Okay, popcorn. So olfactory memory is pretty powerful, very powerful. And emotion. Olfactory system is also very much related to the emotion. You know that a small baby, infant, for example, is crying in his sleep, during his sleep. If you put the baby in mother's lap, the baby will smell mother's body and stop crying. Okay, so olfaction is very much related to, strongly related to the memory as well as emotion. Okay? Many creatures, their survival, their existence depend on their olfactory system. Okay, so just that's all about the olfactory system. Very short. Now we'll talk about the gustatory system. As I have mentioned, that all sensory systems have peripheral organs and central part, peripheral part and central part. In case of gustatory system or taste system, the peripheral organ is the tongue. Your tongue is the peripheral organ for gustation. And in your tongue, you have a number of tiny structures. Those tiny structures you can see with naked eye. If you look carefully, you'll be able to see. Those tiny structures are called papilla. So, in your tongue, you have many tiny structures. Those are called papilla. And if you open the papilla and see inside, you will see the taste buds. So, inside the papilla, you have taste buds. And if you open the taste buds and see inside, you will see the taste receptors. So, it is like this. Let me write it here. So, first, the largest organ is the tongue. Now, in the tongue, you have many tiny structures. Those are called papilla. Those you can see from outside. You can see the papilla from outside. Although they are tiny, but you can see. Now, if you open the papilla, inside the papilla, you will see taste buds. Taste buds are like oranges. If you peel the skin of the orange, you will see slices inside. And those slices are taste receptors. So, tongue, in the tongue you have papilla, and inside the papilla you have taste buds, and inside the taste buds you have taste receptors. You can see the tongue from outside, you can see papilla from outside, but you don't see taste buds and taste receptors because they are located inside the papilla. Now, 4 different types of papilla are present in the tongue. Yes?
So when you burn your tongue,
Yes.
you burn out the papilla?
You do. The receptors die. So, not only burn, when we eat very hard food, some friction occurs, so taste receptors die. And that's why we need the basal cells there. That cell can produce new cells. Okay? So 4 different types of papilla are present in the tongue. Fungiform papilla, foliate papilla, circumvallate papilla, and another type of papilla that has not been seen here, that is called filiform. Just write it down. Filiform is another type of papilla. So fungiform, foliate, circumvallate, and filiform. Fungiform papilla are tiny dot like papilla, very small dot like papilla, and scattered everywhere in the tongue. So you'll see those dots everywhere in the tongue. Those are fungiform. Foliate papilla are like folds. You see the foldings, and are located in both sides of the back part of the tongue. So foliate papilla, they look like folds and located in both sides of the back part of the tongue. And circumvallate papilla are larger round structures and few in number. How many if I ask? 10 to 16, okay, in number, so not many, and they form inverted V, inverted V in the dorsal part or back part of the tongue. So they are located all the way in the back part of the tongue and form inverted V-shaped structure. So those are circumvallate. So if I ask you where you will find which type of papilla, just remember that. Whatever I have mentioned, I may ask you. Okay. Now, if you open the papilla, and see inside, you see taste buds. And I have mentioned that taste buds look like oranges. Although, of course, not that big. Oranges, that very big. It's microscopic structure, very small. But if you see under the microscope, taste buds look like oranges. That means what? Orange has a skin, and if you take off the skin, inside the orange you will find slices, right? Segments or slices. So taste bud is like an orange, and if you open the taste bud, you see many slices inside, and those slices are called the gustatory receptor cells. So those are the receptor cells. You see, this is the taste bud, and these are the slices, like structures. These are the gustatory receptor cells. Those are the main cells, and you will also see basal cells inside the taste bud. And basal cells are those blue cells. You see small blue cells. Those are the basal cells. And you already know that basal cells are stem cells, and they can produce new receptor cells. That is very important, as you have asked that, when you burn your tongue or you eat very hard food, then what can happen? Actually, it happens all the time. When you drink coffee, some receptor cells die, and that's why you need the stem cells, the basal cells there. They can quickly replace the dead cells with new cells. Okay, so those are two types of cells you see inside the olfactory, gustatory buds, taste buds. Now, this is the surface of the tongue, okay? And in the surface of the tongue, you have many tiny pores or holes on the surface of the tongue. And why those taste pores are there? This is the surface of the tongue, so this is the pore. When you eat the food, what happens? Salivary secretion occurs. You know that secretion of saliva occurs, and the food molecules get mixed with saliva, that watery secretion, highly watery secretion. And that is very important, because if the food molecules don't mix with water, saliva, then it will not enter through the taste pore and will not reach to the receptors, because receptors are here. So the food molecules must go inside to get attached to the receptors here. Okay? So that's why your food must get dissolved in saliva. Otherwise, dry food will not be able to go in. Liquid food will easily go inside through the taste pores. That makes sense. And that's why if you eat something very dry, first few seconds you won't taste anything. If you put something very dry on your tongue, you won't taste it. When the salivary secretion will occur, then you will start to taste the food. Because if the food molecules are too dry, they are not able to go in. So the liquid food will go in and will bind to the receptors. Okay? So that's why the pores are there. Okay, so we'll see what happens. When the receptors bind with the food molecules, then the signal will start to travel to the brain structures. First, the signal, the taste signal, will be taken to the medulla oblongata in your brain stem. And in your medulla oblongata, you have a structure that will receive the taste signal. That structure is called solitary nucleus. You see these green structures in the medulla oblongata. These are solitary nucleus, and they will receive the signal from the tongue, from the receptor. So remember that solitary nucleus. If I ask you in the test that which structure will receive the taste signal from the tongue, that's the solitary nucleus. Solitary nucleus is located where? In the medulla oblongata of the brain stem. Okay. Now, which nerves carry the signal from the tongue to the solitary nucleus? Two nerves. One is facial, that's the cranial nerve number 7, and another is glossopharyngeal, that's the cranial nerve number 9. So facial and glossopharyngeal, those two nerves take the taste signal from the tongue to the solitary nucleus. And then the signal from the solitary nucleus will go to the thalamus. And you know that thalamus is called the major sensory relay station, and almost all sensory signals will go there. So thalamus will receive the signal, and from the thalamus the signal will go to the gustatory cortex. That's the highest area. So this is the gustatory pathway or taste pathway. So from here, in this part, I may ask you following questions. Important, of course, others I can ask. From the largest to the smallest, okay? How the receptors are located. Tongue, then papilla. Inside the papilla, you have taste buds. Inside the taste buds, you will see the slices. Those are the taste receptors. Okay, and then after the taste receptors get activated, the signal is taken where? To the from the tongue to the solitary nucleus in the medulla oblongata. And which nerves carry that signal? Two nerves, glossopharyngeal and facial. Okay, remember that. And from there it will go to the thalamus. From thalamus it will go to the gustatory cortex. That's the pathway. Okay. Okay. Now, we'll talk about the auditory system. Auditory system is responsible for hearing the sound, and it also has two parts. The peripheral organ of the auditory system are the ears. You have two ears, and those are the peripheral organs. And the ear has three parts: external ear, middle ear, and internal or inner ear. So the ear has three parts. Remember, only a part of the external ear we can see from outside. Most part of the ear is located inside that we don't see. Only a part of the external ear we can see from outside, even not the whole external ear. And of course, the middle ear and inner ear, those parts are located inside, so we don't see from outside. Okay. So if you see those three parts in this picture, external ear is this part, and external ear is separated from the middle ear. This is the middle ear part by a membrane, round membranous structure that is called the eardrum or tympanic membrane. So tympanic membrane separates the external and middle ear. It is the partition. It is also called the eardrum. Now, in some books, you will see that they have included included the tympanic membrane as a part of external ear. That is also fine. But if I ask you, just say that it separates external and middle ear. It is the partition between external and the middle ear. Now, important parts of the external ear. Pinna is the largest part that you can see from outside. That's the pinna of the ear. And lobule is the lowest soft structure of the ear. So this is the pinna, also called auricle, and this is the lobule. Okay? And this is another part of the ear, that is the external acoustic meatus. You know that meatus is canal-like structure. So the external acoustic meatus is the outer ear canal. Sometimes we also say outer ear canal or external acoustic meatus. Same thing. Okay, meatus is the canal. Then, what's the function of the external ear? Pinna, you will see in some animals, like for example elephants, their pinna is very big. Pinna captures the sound. Okay, so pinna captures the sound and channels the sound to the inner part of the ear through the external acoustic meatus. So pinna captures the sound and channels the sound to the inner part of the ear through the external acoustic meatus. It's like, the funnel you use to pour the, oil or liquid in something. So its funnel part is the pinna, and the tube part is the external acoustic meatus. That's the function of the external ear, capturing the sound and channeling it towards the inner part. Then here is the tympanic membrane or eardrum. and this is the middle ear. Middle ear is a cavity. Middle ear is a cavity. It has two things. Inside the middle ear cavity, you will find two things. Tiny bones, those are called ossicles, and two tiny muscles. So I am repeating again. In the middle ear is what? It is a cavity. It contains what? Three ossicles or tiny bones and two tiny muscles. You don't see muscles here, you see three tiny ossicles in this picture. So we'll see in next picture the name of those three ossicles: malleus, incus, stapes. And then this is the inner ear. Inner ear is the most important part of the ear. It has very important structures in it. Which important structures are present in the inner ear? Two important structures. One is called the cochlea. Cochlea is a snail-like hard structure. Cochlea is a snail-like hard coiled structure. It is a very important structure in your ear. I'll say the most important structure, because inside the cochlea you have the sound receptors. So now you can imagine that if the cochlea is destroyed, then the sound receptors will be destroyed, the person will not be able to hear. So that's why I said that it is the most important structure, the cochlea, which is located in the inner ear. What type of structure is it? It is a coiled, snail-like hard structure. It contains what? The sound receptors. Okay? So that is one important structure. And another, so this is the cochlea. This is snail-like hard structure that contains the sound receptor. And another structure is this. This structure has two parts. You see the tube-like structures. These are called semicircular canals. Semicircular canals. Those tube-like structures. Semi is not complete, circular. Okay, it's not incomplete circle-like structures. And those semicircular canals are attached to a part, attached to this part. This is called vestibule. So I am writing here the tube-like structures, those are called semicircular canals, and those tubes are attached to the part that is called vestibule. Okay? And these two structures, just remember, are responsible for balance of the body. These two structures are related or responsible for the balance of the body. So you see, completely two different functions are performed by two parts of the inner ear, or two structures in the inner ear. Cochlea is responsible for the processing of sound. This is one type of signal. And the semicircular canal and vestibule, they are responsible for balance of the body, which has nothing to do with the sound. So in the inner ear, we have, inner ear has two functions. We have two types of structures, and they perform completely two different functions. Yes?
For the middle ear, do you want us to know the names of the bones
Yes.
there, or just know?
So the bones, yes, you need to know. Very important, okay? So the bones or ossicles in the middle ear are how many there? 3. The first one is attached to the eardrum or tympanic membrane. That one is called the malleus, which is attached to the tympanic membrane. And then the middle one is called the incus. And the last one or innermost one is called steps. Some people say stapes, which is fine. Steps. So malleus, incus, and steps. And you see those 3 here, ossicles, and they are attached to each other. So this is the malleus. One end is attached to the tympanic membrane, inner side of the tympanic membrane, and the other end is attached to the malleus, incus. And incus is attached to the steps here. And steps is attached to the oval window. So this part of steps is attached to the oval window. So just remember, oval window is nothing but a membrane. It's a membrane, oval shaped membrane. So now those are the 3 ossicles. And 2 muscles are, 2 tiny muscles are present there too. Those are tensor tympani and another is stapedius. Okay, so I think no, this is not right. Stapedius. Okay, so these are the 2 tiny muscles. Okay, so 3 ossicles and 2 tiny muscles. Now, remember here this one because I continue from here. So 3 ossicles are attached to each other, and the last one, stapes, is attached to the oval window, which is a membrane. So if this is the stapes or steps, it is attached to the oval window like this. Okay? Just remember that. Now we'll see the structure of the cochlea, where the sound receptors are located. We need to see where the sound receptors are located, because receptors are the most important parts. Okay. Now cochlea is a coiled, snail-like structure like this. Okay. So what we can do, we can make it straight. Since it is a coiled structure, we can hold the end and pull it like this. You can actually do very tiny structure like this. You can see under the microscope. It's a coiled structure. You use fine forceps and make it straight. Like this. Okay. Make it straight. Okay. And after that, if you see inside, you see 2 partitions are present inside the cochlea. So after you make it straight, this is the cochlea, and if you see inside, you'll see 2 partitions by 2 membranes separating inside part of the cochlea. So since 2 partitions, now how many cavities you got? How many chambers? 3. Very good. So let's cut like this. Make a section like this and see. So you'll see like this. This is the whole inside the cochlea, and 2 membranes and 3 cavities, okay, inside the cochlea. And we'll see that here. You see those 2 tiny muscles, tensor tympani and stapedius. Okay. Here. So this is a section of cochlea like this. You take a chunk of cochlea, cut like this, and see. You see 3 cavities. and those cavities are separated by two partitions or membranes. The upper one is called the vestibular membrane, this one. Vestibular membrane. Okay? And the lower one is called the basilar membrane. So this is the basilar membrane. Okay? Basilar membrane. So those are the two membranes or partitions. Vestibular and basilar. Now, on the basilar membrane, you have a structure. On the basilar membrane, you have a structure that is called the organ of Corti. Organ of Corti. Here it says spiral organ of Corti. Just know that it is called the organ of Corti, sitting on the basilar membrane. Okay, sitting on the basilar membrane. This is a very important structure because your hair cells, the receptor cells are called hair cells, sound receptor cells are located inside the organ of Corti. So these are the hair cells. Okay. There are two different types of hair cells we see. Just know that. These are the hair cells. These are the sound receptor cells. And why these are called hair cells? Because they have hair on the apical end. Free end has hair. So these are the hair cells. These are the sound receptor cells. These are the sound receptor cells. And two types of hair cells we see there in the organ of Corti, outer and inner. So two groups of hair cells, outer hair cells and inner hair cells. Okay. Probably you will see next week. Here you see inner hair cell and outer hair cell inside the organ of Corti. Okay. Now these cavities are filled with fluid. So we have fluid everywhere. Fluid. So these cavities are fluid-filled cavities. So cochlea is a fluid-filled, snail-like coiled structure. Now I have added another word there. Cochlea is a fluid-filled, snail-like, hard coiled structure that contains the sound receptor cells or hair cells. That's why the cochlea is the most important organ. If you destroy the cochlea, if you destroy the organ of Corti, then the person will not be able to hear because the hair cells are the sound cells. There, receptor cells. Now, you remember I said that stapes, the last ossicle, is attached to what? Oval window. Remember that? Oval window? I told you to remember that. Steps is attached to the oval window like this. And oval window is what? A membrane. Okay? So now, where the oval window is located? Oval window is here. That membrane is here. This is the oval window attached to the cochlea. And here is your steps, that tiny ossicle. Okay? So what happens see here, sound waves enter into your ear. Okay, sound waves one after another. Sound is like this. Sound is sinusoidal wave like this. That means one after another. They're coming one after another like this. And entering, those waves are entering into your ear. Yeah. And first the waves will hit the eardrum or tympanic membrane. Will hit here. One after another like this. Will hit like this. And your tympanic membrane will move like this because it is a thin membrane, eardrum or tympanic membrane. So sound waves will hit one after another and it will move like this. And here you have the first ossicle attached to the inner surface of the tympanic membrane. That's the malleus. And you know that those three ossicles are attached to each other. So what will happen? They will start to shake like this. Those three ossicles, okay, will start to move like this. Why that movement is important? Because just know that movement of the ossicles will amplify the pressure, increase the pressure. Okay, so that's the function of the ossicles, will increase the pressure. Anyway, so those three ossicles will start to move like this. And the last ossicle is the steps. Right? So this one will also move like this. Steps will also move like this. And that will push the oval window because it is a membrane. Oval window, oval shaped membrane will move in and out like this because it is going like this, in and out. And here you have fluid everywhere because those three cavities are filled with fluid. Fluid, right? So when the oval window will move like this, it will push the fluid in and out this way. Okay? So the waves will be created in the fluid. Because if you push the fluid like this, wave will be created. And that wave will do what? Will push the basilar membrane, this membrane, basilar membrane, up and down, like this. Okay? This fluid, wave in the fluid, will push the basilar membrane up and down. Okay. And we are almost done. So on the hair cells, you have another structure here. Another soft jelly-like membranous structure. Soft jelly-like soft membranous structure that is called tectorial. Tectorial membrane. Okay? And what happens? The tectorial membrane is what kind of membrane? What kind of membrane? Tectorials. Soft? Soft what? Jelly-like. Okay. So here are your hair cells. These are the hair cells, right? So these are hair cells. Just think that these are hair cells. And here is your tectorial membrane on the hair cells. And the hair of hair cells will enter, will be inserted in the tectorial membrane since the tectorial membrane is soft. The hair are inserted in the tectorial membrane. And here you have the basilar membrane at the bottom of the hair cells because hair cells rest on the basilar membrane. Okay? So this is the basilar membrane here and this is the tectorial membrane. And when the basilar membrane will move up and down like this due to that wave in the fluid, what will happen? It will push the hair cells up and down against the tectorial membrane. And when it will push the hair cells up, what will happen? The hair will bend. Does it make sense? The hair will bend. and when it will move down, the hair will again get straight. Bend, straight, bend, straight. That will continuously occur one after another. Okay? And that bending of the hair will activate the hair cells. So that bending of the hair will activate the hair cells. So it's a mechanical kind of activation due to pressure. That pressure is bending the hair, and that is activating the hair cells. Okay? So it's nice, to know everything how it's getting activated. So here you see the tectorial membrane. This is the tectorial membrane. These are the hair cells. Okay? And this is the basilar membrane. So basilar membrane will move up and down like this, and here are inserted in the tectorial membrane. So the hair will get bent and straight like this. Okay? And that will activate the hair cells, and signal will start to travel, okay, towards the brain. Which nerve will carry the signal from the hair cells to the brain? That nerve is the auditory nerve. Auditory nerve, of course, auditory nerve will carry that signal because it is auditory signal. And auditory nerve is 8th cranial nerve number 8. So remember that. I may ask you that auditory nerve is cranial nerve number what? Cranial nerve number 8. That will take the signal from the hair cells to the brain. Okay, so you now know the mechanisms of the activation of the receptors. Yes?
Do we need to know the scala vestibuli, scala medium, scala tentacle?
Those 3 cavities?
Yeah.
Yes, no, yes, no? Or do you want?
I don't know, you just didn't really mention those, I don't know. If you
Yeah,
need
so those 3 cavities, just know the names. If I ask you, the question will be like this: which one is not the cavity inside the cochlea? So if you just remember, you will know which one is not. Okay. Okay. So from here, important things are, the parts of the ear, external, middle ear, inner ear, which structures are located in which part, and organ of Corti is what? It is a structure that contains the hair cells located on the basilar membrane, right? And which 2 membranes separate inside the cochlea into 3 chambers or cavities: vestibular membrane and basilar membrane, right? And the organ of Corti is located on the basilar membrane. What is tectorial membrane? I said that it is a soft jelly-like membrane that lies on the hair cells, and hair of the hair cells are inserted, right? And 3 ossicles, 2 muscles, right? If I ask you which structures are related to the balance and which one is the most important structure that contains the receptors, that's the cochlea. Snail-like, hard, fluid-filled, coiled structure is the cochlea located in the inner ear. Okay. So those things I have mentioned, I will ask those as your questions in the exams. Okay. So just try to, you have taken the note, you should be able to answer. And auditory nerve is cranial nerve number what? You see the auditory nerve is connected to the hair cells. So from the hair cells, when the hair cells will get activated by the bending of the hair, the signal will start to travel through the nerve. You see the branches of auditory nerve are connected to the hair cells, right? And you see the hair, the apical end of the cells are attached to the tectorial membrane. If you see the hair cells under the microscope, you can easily tell which are the inner and which are the outer hair cells because they are arranged in 2 different ways. Inner hair cells are present in straight line we say linear arrangement. And outer hair cells form U or V-shaped arrangement. So it is easy to say which are the inner hair cells, which are the outer hair cells. Inner hair cells form a line, linear, and outer hair cells form U or V-shaped arrangement. Okay. Probably I won't ask you the structures, name of the structures of central auditory pathway. Just know that you know that all sensory systems have the peripheral part, and in case of auditory, peripheral part is the ear or the ears, and we have talked in detail about the ear, structure of the ear, and how the cells get activated. Now the auditory nerve will take the signal to higher brain structures. This is the central part. This is the peripheral part. This is the central part. First, the signal will go to the cochlear nuclei in the medulla oblongata. From there, it will go to the inferior colliculus. You have seen superior and inferior collicula. You remember when you did the dissection of the brain? I showed you upper two round and lower two smaller round structures called corpora quadrigemina. Upper two are superior colliculi, lower two are inferior colliculi. So lower two will receive the auditory signal. Now you see you can connect that. And from there, the signal will definitely go to the thalamus because it is the major sensory relay station. And from there, it will go to the auditory cortex, primary auditory cortex you already know. And the upper part of the temporal lobe, you will remember that green area in your brain model. That's the primary auditory cortex. So that's the highest area where the signal will finally arrive. Okay. So that's the central auditory pathway. Okay. Hearing loss. Two types of hearing loss can occur. One is called conductive hearing loss, and another type is called sensory neural hearing loss. Conductive hearing loss is very easy to understand. For example, if anything in your ear blocks the sound waves from entering into the inner ear. For example, if I put a piece of cotton in your ear, that will create conductive hearing loss because the sound waves will not be able to go to the inner ear, right? Make sense? If I put a piece of cotton in your ear, that's conductive hearing loss. If a tumor grows in your ear, then the sound waves will not be able to go to the inner part. So tumor can block the sound wave from entering. So that will cause conductive hearing loss. So sometimes wax, that mix with the dirt particles that enter into the air and may form solid structure that can block the sound from entering into the ear. So that oily secretion can also block the ear. So those are examples of conductive hearing loss, and easy to fix. If you just remove the tumor, if you remove the wax or remove anything that is blocking the ear, then the patient will be fine, will feel okay. So that's the conductive hearing loss. Sensory neural hearing loss is almost impossible to fix. The reason is in sensory neural hearing loss, what happens, the hair cells are destroyed. So those hair cells are destroyed. Now, the problem is in your organ of Corti, you don't have the stem cells, you don't have the basal cells. You remember in your tongue, in your nose, you have basal cells. Those stem cells can multiply, right? But in your ear, in the organ of Corti, you don't have the stem cells there. So once the hair cells die or are destroyed, then no new hair cells will be produced. And that's why if sensory neural hearing loss occurs, then no new hair cells will be produced. So the person suffers from hearing loss rest of the life. Now we got some devices that can improve the situation. What we do, cochlear implant, you have heard probably all of you. What we do, since the hair cells are destroyed, no signal is going to the nerve, we can connect a wire here directly, bypassing the hair cells, this part. We can, from outside, we can activate the nerve through a wire and pass the electrical activity that can activate the nerve, can send the signal bypassing the organ of Corti or hair cells. That's what is done in cochlear implant. They connect the outside directly to the auditory nerve fibers. Okay. That can improve the situation. Now, why or how the sensory neural hearing loss can occur? There are few most common causes are, number one, prolonged exposure to high frequency sound. If someone works in a factory that produces high frequency sound, and if the person works 12 hours every day, then the chance of sensory neural hearing loss is very high. Okay? So that is one. Prolonged exposure to high frequency sound. Number two, high dose of powerful antibiotics. If you give very powerful antibiotics to